Good morning everyone, I am Wilva Mana and today we are going to discuss the processes of sexual differentiation and explain some of the disorders that can occur in this process. But first, let us define sexual differentiation. So what is sexual differentiation? So it is the process by which the male and female sexual organs develop from neutral embryonic structures. The normal human fetus of either sex has the potential to develop either male or female organs, depending on genetic and hormonal influences. Now let us proceed to the process of sex differentiation. We all know that male has an XY chromosomes, while female has XX chromosomes. One is passed down from mom's sex, which is the X chromosome, and one from daddy's sperm, which is either X or Y chromosomes. The factors that determine our sex begins in earliest fertilization. If the sperm is carry an X chromosome and fertilizes the egg, a girl will be conceived. While if the sperm carry an Y chromosome and fertilizes the egg, a boy will be conceived. So to make sense of it, let's go back to biology and discuss it more. In the beginning, every embryo has the potential to form both male and female genitalia. The organs will eventually differentiate to become either male or female sex organs. That means that all our sex organs come from the same foundations. The phallus will develop either a penis or clitoris, and the genital folds will develop either scrotum or labia, while the gonads will develop either testes or ovaries. Undifferentiated or genderless zygote passes both rudimentary male structures called the Wolffian ducts and the female rudimentary or primary structure called the Mullerian ducts. Whether the sex structure develop into male or female, it is dependent on the presence or absence of the two hormones. These hormones are called the testosterone and Mullerian inhibiting substance, abbreviated MIS. So this is the internal structure of undifferentiated zygote. As I said, it passes both Mullerian and Wolffian ducts. Testosterone will act in the Wolffian ducts to promote the development of male reproductive structures, while the MIS will act in the Mullerian ducts to prevent the development of female organs, and it will develop in the epididymis, vas deferens, seminal vesicle, and ejaculatory ducts. So in other words, the presence of testosterone and MIS will cause the fetus to develop into a male. Now, let's look at this to the different angle. So if testosterone is absent, this will prevent the Wolffian ducts from developing into male reproductive structures. Furthermore, if the MIS is absent, this will stimulate the Mullerian ducts to promote the female reproductive structures, and it will develop in the fallopian tubes, the uterus, and the upper part of the vagina. So in other words, the absence of the testosterone and MIS will cause the fetus to develop into a female. So now let's talk about the SRY gene. It is found in the Y chromosome that triggers the embryonic development as a male. The process begins at week 7. It activates the signaling productions of the male and makes the gonads into testes. The testes secretes the male hormones, which is the testosterone, to produce AMH or anti-Mullerian hormone. So the testosterone will cause the phallus to develop penis. So like what I've said a while ago, that the Wolffian ducts will be formed in the epididymis, vas deferens, seminal vesicle, and ejaculatory ducts. So since the Mullerian ducts is for female, it will degenerate since the AMH stops the uterus and fallopian tubes in developing. On the other hand, female does not have the SRY gene, so that gonads will turn into ovaries without testosterone and AMH, and the female organs will develop and the Wolffian ducts will be degenerate. Changes of any of these processes can cause disorders of sex development, or the DSDs, or termed as the intersex conditions. Disorder of sex differentiation, or DSD people, are born with sex characteristics that do not fit typical binary notions of male or female. Now, Let us see what are the different types of DSD that can occur in the process of sex development and the causes of it. The first one is the sex chromosome DSD. The most common sex chromosome compositions are XX and XY, but there are several possible compositions, for example, XO, XXY, XXX, XYY, XXYY, XXXY, etc. and etc. If only XX is female and only XY is male, then how do we account for it to be called chromosomes? Their sex organs are normally formed as either male or female. Those children that has this kind of disorder may not go through normal sex development at puberty. For example, a child with female sex organs may not start having periods and may have small breasts. The second one is the mixed genitals and sex organs. This is a very rare type of DSD where the child has tissue from both ovaries and testicles. The gonads can be any combination of ovary, testes, and ovotestis. Just combine the ovary and the testes. Without fully functioning gonads, it will not produce hormones. That's why the body did not develop in typical male or female pattern. Children with this type of DSD have female chromosomes. Although the cause of this condition is not known, some cases have been linked to genetic material normally found on the Y chromosome that is misplaced on the X chromosome. The third one is the Rokitansky syndrome. Some females are born without a womb, cervix, and upper vagina. Some may have underdeveloped organs. In this condition, the ovaries and external genitalia or vulva are still present. They will still develop breasts and pubic hair as they get older. MRKH, or also known as Mayer-Rokitansky-Hauser have normal female structure, but the Mullerian ducts fail to develop completely, resulting in a missing uterus. The first sign of Rokitansky syndrome is that a girl does not start having periods. Sex may also be difficult because the vagina is shorter than normal. Women with Rokitansky syndrome who have no womb cannot become pregnant, but it is sometimes possible to take eggs from individuals to make surrogate pregnant.